we will be studying two slides: bone marrow aspiration smear, and then the other one is the bone marrow tissue. Now, the more difficult slide to study is the bone marrow aspiration. So that slide, the smear is used to identify the cells belonging to the lineage in the production of the mature blood cells. Now, there are two cellular lineages that can be identified in a bone marrow aspiration smear. So, namely, the myeloid and then the erythroid series. So, we will begin characterizing the cells belonging to the erythroid series. Now, we all know, as discussed in your lecture, the first cell in the production involved in the production of blood cells are the pluripotential stem cells. When we say pluripotential, they have the capability to produce all types of blood cells. Okay? And then, when a cell becomes committed to produce one particular type of blood cell in that line of production, the first identifiable cell for the production, first identifiable stem cell for the production of the red cells will be that cell, a pro-erythroblast. Now, what are the identifying features? So you will be made to identify this in your slides later. What are the identifying features of a pro-erythroblast? Pro-erythroblast is the largest cell in the erythroid lineage. So, usually, cells that will produce red blood cells in the bone marrow are clustered together. And then cells which are destined to become white blood cells, their stem cells in the bone marrow are also clustered together in the bone marrow cavity. So that is why we will be able to see the precursors of the red cells amongst each other. Now, among the precursors, identifiable precursors of the red blood cells, the largest will be, as you can notice, according to their size, is the pro-erythroblast. Now, what are the morphologic characteristics of the pro-erythroblast? Notice the color of the cytoplasm of the pro-erythroblast. It is basophilic, dark blue or dark violet, and then it will also have a very large nucleus. And then ang ginagamit ko in identifying the erythroid series cells, not is the characteristic of the nucleus. The nucleus of the cells producing red cells is usually dark, and then you will be able to see parang mga butas-butas. So dark, and then there are holes in the large dark nucleus. That cell usually belongs to the erythroid series. So this cell will have a large nucleus with coarse chromatin pattern or dark nucleus with nuclear holes. And then the next cell in the series is the basophilic normoblast. Now, basophilic because of the color of its cytoplasm. Very intensely basophilic. So, pag basophilic ang isang component ng cell, it will appear dark blue or dark violet. Now, what is the reason behind the color of the cytoplasm of this cell? Okay? A mature red blood cell, in order to perform its function, must have what type of protein in its cytoplasm? Hemoglobin. Hemoglobin is a protein. What are the organelles involved in the synthesis of hemoglobin? Well, the ribosomes, particularly the rough endoplasmic reticulum, and you need a command coming from the nucleus, which will come in the form of mRNA, rRNA, and then after being synthesized in the rough endoplasmic reticulum, hemoglobin will be packaged in what organelle? Golgi apparatus, Golgi complex. So that intensely blue color of the cytoplasm is secondary to the presence of the organelles involved in the synthesis of hemoglobin. And then the next cell in this, by the way, you may call this cell as erythroblast also. So normoblast, erythroblast, same banana. Next cell in the lineage is the polychromatophilic normoblast. So poly is Greek for many, and then chromo for color. So this cell will display two colors in its cytoplasm. So because in this lineage of development of red cells, the cytoplasm will already begin to synthesize hemoglobin. There is already hemoglobin in the cytoplasm imparting pinkish hue in the cytoplasm, plus the presence of the rough endoplasmic reticulum and then the RNA. Okay, the RNA will have basophilic color, and then hemoglobin will have the pink color, so polychromatophilic. And then next cell in the lineage is the orthochromatophilic normoblast. Now, siyang orthochromatophilic because ortho is Greek for correct or right, and then chroma color. So, now siyang orthochromatophilic normoblast. These are the mature red blood cells. Ayan sila. The color of its cytoplasm is almost pink already, as pink as that of a mature red blood cell. Okay, pink is the color of a mature red blood cell cytoplasm, thus it is called an orthochromatophilic normoblast. So, however, the nucleus is still present. And then eventually, the nucleus will be extruded from the cytoplasm. So, next cell in the lineage is a reticulocyte. Who among you here are medical technologists? Dito. Yung medtech dito. Anong stain ang ginagamit to identify retics? Vital stain. Brilliant crescent blue. You will be able to see blue granules in its cytoplasm. These granules will represent the remnants of the mRNA and the rough endoplasmic reticulum in the cytoplasm, indicating that there is still ongoing hemoglobin synthesis. And then, this is the cell that will be released normally from the bone marrow. All these cells will stay in the bone marrow. So unless there is a need to release more number of red cells in the circulation. So, and then after 1 to 2 days, there will be completion of hemoglobin synthesis. That is why the color of the cytoplasm will become pink already. Okay? So, what is the reason behind the loss of the nucleus in the red cells? Aside from that, okay, so now for the specific features of each cell in the lineage. So that's a pronormoblast, so basophilic cytoplasm with pale perinuclear area. So basophilic cytoplasm with pale perinuclear area. And then some of the erythroblasts will display these protrusions, ear-like protrusions, imparting a Mickey Mouse-like appearance.
Mickey Mouse.
So, no other cell in the bone marrow will display these protrusions. So, if you will be able to see that in your smear, it should be the proerythroblast already. And then, okay, following the proerythroblast, okay, is the basophilic normoblast. We have already discussed the identifying feature of this cell. The cytoplasm is intensely blue or violet in color. So that's the name, basophilic. And then the next cell in the lineage, the polychromatophilic normoblast. So in a better stained smear, you will be able to see blue and pink colors in the cytoplasm of a polychromatophilic erythroblast or normoblast. And then next is the orthochromatic or the orthochromatophilic normoblast. These are the mature red cells. Notice that the color of their cytoplasm is almost similar with the cytoplasm of these four orthochromatophilic normoblasts. Their difference will come only in the presence of the nucleus. And then this cell will require vital staining in order to be identified. So you will not be able to see them in your smears. And then that should be the regular appearance of a mature red blood cells, which we have already discussed last time. Now, for the myeloid series. So these cells, this series will produce the neutrophils, the basophils, and the eosinophils. Now, the earliest identifiable cell in this series are the myeloblasts. Now, notice the color of the cytoplasm or the nucleus of that cell. Okay? And then compare the characteristic appearance of the nucleus of that cell with the characteristics of the nucleus of the cells belonging to the erythroid series. Mas dark ang color ng nucleus, and then mayroong mga butas-butas which are absent in the myeloid series. Okay? Myeloblast will have a very big nucleus with scant cytoplasm. Big nucleus, scant cytoplasm. And then, you will be able to see 2 to 5 nucleoli. Now, usually, ang cells ng katawan natin, dark ang color ng nucleolus. In the case of the precursor cells, precursors of the blood cells, light ang color ng nucleoli nila. So, 1, 2, 3. That cell is having 3 nucleoli. And then, after the myeloblast follows the promyelocyte. How will you be able to identify a promyelocyte? Notice the dirty appearance of the cytoplasm. In this stage of leukocytic development, the first type of granules will appear in the cytoplasm. What is the first type of granules? Okay, the azurophilic granules. So, the azurophilic granules will appear violet or blue. Kaya, we are able to see violet granules in the cytoplasm. That should be the promyelocyte. And then the next cell in the lineage is the myelocyte. Now, kung ang promyelocyte mayroong azurophilic granules, pagdating sa myelocyte, the azurophilic granules will be accompanied already by the specific granules. Okay? Kaya mayroong blue granules, mayroon din pink granules, which are smaller than the blue granules. And then, there is a clear area immediately adjacent to the nucleus. That zone or area is called the Golgi zone. Okay? The Golgi apparatus area. And then the next cell in the lineage is the metamyelocyte. Okay? The contents of the cytoplasm of the metamyelocyte is similar with that of the myelocyte, containing both azurophilic and specific granules. The difference will come in the shape of the nucleus. Okay? There will be indentation already in the nucleus of a metamyelocyte. Okay? That's the only difference, the shape of the nucleus. Next cell in the lineage after the metamyelocyte is the bond cell. Okay, wherein there is already elongation of the nucleus. So, and then the mature form will be the segmented granulocytes. Neutrophils, notice that there are already segmentations in the nucleus. So, okay, there is an erythroid cell present. Identify that cell. The color of the cytoplasm. Anong makita ninyong color sa cytoplasm? Violet and pink. So that it should be called a polychromatophilic normoblast. Okay. So that's a myeloblast. Notice there are no granules in the cytoplasm. The nucleus is very large, and then the volume of cytoplasm is scant. Next cell in the lineage. Okay, that's a better picture of a myeloblast. Large nucleus, scant cytoplasm without granules. Those are the nucleoli. Next cell in the lineage is the promyelocyte. If the pronormoblast is the largest in the erythroid, the largest in the myeloid is the promyelocyte. Okay? That's a myeloblast. Notice that in this field, we are seeing majority are myeloid cells because they are clustered together. So there are already azurophilic granules in the cytoplasm. Okay, so those are the azurophilic granules in a promyelocyte. Okay. Next cell in the series. At this stage of development, we can already identify the precursors for the neutrophils, eosinophils, and basophils. Kasi magsisilabasan ng specific granules in their cytoplasm. Aside from the blue or violet-colored azurophilic, there will be appearance already of the pink-colored specific granules. Okay? Ayan. Blue and then pink granules na makikita sa cytoplasm. Then how do you call that zone? Golden zone. And then, okay, that should be the myelocyte for an eosinophil. Notice the presence of the larger bright pink to orange granules. And then basophilic myelocyte, malalaki ang granules. So, covering the outline of the nucleus. Then next cell in the series, the bond cell. That's a metamyelocyte. Okay, metamyelocyte. And then next is the bond cell. Okay. Okay, that's another appearance of a bond cell. Notice the difference of a bond cell with that of a mature or segmented neutrophil. Do you have a bone marrow aspiration biopsy smear in your box? Would you like to have a copy of this presentation?
Yes.
I-paste ko na lang sa ano. Ay, paste ka na lang sa desktop mo. Sa documents na hematopoiesis ang finding.
Flower body. Flower, no?